check my email so I can say that once in my life I checked my email in the middle of a seminar. It'd be a little too addictive, I think. Is everyone here? Almost.
The important people are here.
Important
Yeah,
people. Well, I must say that I have never experienced such enthusiasm from a group of students. So it's just such a tribute, I think, to yourselves and your mentors and the education you're getting here. It's really just wonderful, the intellectual engagement and the critical thinking. So whatever's happening here at your institute that's prompting you to be so enthusiastic about science, I think it's just absolutely wonderful. But we've got to get through this talk. And I do want to finish before lunch. Now you might be wondering, when I talk about these microtubule polarity patterns, well, how do you know? Has anyone even considered that? How do you know? How do you know what pattern? If you do electron microscopy, if you do immunofluorescence, you wouldn't know the microtubule polarity orientation. And one of the reasons why this was a special discovery that we made in 1988 with dendrites is that the technique is very tedious and the sort of thing that most people didn't even want to deal with, didn't even want to touch because it was very difficult. And that's another thing I think sometimes as young scientists coming up, don't be afraid of challenges. If there's a very important question out there and that everyone's telling you that's too hard, that's too difficult, don't do that, it'll take you too long, don't necessarily be discouraged by that, because sometimes you can really make very important discoveries. There are so many contributions in science among so many different players, somebody really focusing on the biophysics or somebody hammering the structural biology or whatever it may be, and really learning something that was really not known before. So the way that this was done years ago is a technique that was developed by Steve Heidemann, who was my graduate advisor. And if you just do a kind of a standard microtubule assembly buffer, and you expose microtubules, then it just assembles as it normally would. And what Steve came up with, and he'll tell you, was kind of by accident that he threw together a buffer with a lot of DMSO and some other sort of strange features that the new tubulin added to the sides of the microtubule as this structure is like a hook. It's a little bit like those microtubules in flagella that are kind of have a half microtubule growing off of them. And as long as that hook doesn't close, as long as you get in there and you fix your cells, you have to extract them to let this tubulin in and fix them just on time. If you look in cross-section, they look like these little hooks. And those hooks will reveal the polarity orientation of the microtubule. So if you're looking down on the cross-section and you see a clockwise hook, that means you're looking at the plus end of the microtubule. If you see a counterclockwise hook, it means you're looking at the minus end of the microtubule. Now, if you're working with cultured neurons, the way that I usually do, this is very tedious because in order to cross-section for electron microscopy, you need electron microscopy to be able to see this. You're cross-sectioning a lot of axons, and it's very delicate work, and most people didn't want to venture in that direction. But I did, and we came up with the observations that I just talked about. One of the things that's interesting about the hooking data is that it was always considered that there was a little bit of noise in the system. In other words, if you got 95, 98% clockwise hooks, what that meant is you've got uniform polarity orientation. All the microtubules have their plus and distal. And those couple percentage of counterclockwise hooks, they're probably just noise, just noise in the technique. Well, more recently, there is a much more straightforward method for looking at microtubule polarity orientation. And you're going to hear a lot about these plus tips from Anna when she gives her talk at the workshop. And the way that these plus tips work, these are naturally occurring category of microtubule-related proteins that associate with the plus end of the microtubule only when it's assembling. Only when it's assembling. So that you can know the plus end of the microtubule because you express a fluorescent construct, GFP fluorescent construct for EB1, EB3, class, one of these plus tips. And you will see the microtubules shoot off like comets. And the comet shape will point toward the minus end of the microtubule, the comet head, toward the plus end of the microtubule. And if you do this with cultured neurons, then you see the polarity orientation in a live cell moving. So it's very dramatic, and you can see the comets shooting off toward the tip of the axon, and in dendrites you see a mixture of the comets in both directions. So this is a nice confirmation to be able to visualize what we saw with that very complex method that only can be performed on fixed cells. Here we could see it with live cell imaging. And that was very nice to be able to confirm. One of the things that you start to see with this technique is if you look at the axon, occasionally, you do see a minus end distal microtubule every once in a while. And that was really a very interesting finding because what it suggests now, I don't say uniform, I try to say nearly uniform, because on rare occasion you do see a minus end distal microtubule that kind of sneaks in there. I'm going to talk about that just a minute after I field a couple of questions.
This hook-like structures, they are seen in live cells also? Yes.
No, the hook-like structure is experimental in nature. You extract the neurons with a detergent in the presence of this wacky microtubule buffer, and you have exogenous tubulin that you got from a cow brain or a pig brain, and they assemble onto the native microtubule. So the hook is not natural. The hook is something you're creating experimentally to reveal the polarity orientation.
So when tubulin is isolated from bovine brain or goat brain, that means high concentration of glutamate. So does that also induce such kind of hook formation or not?
Well, as long as the buffer finally at the end of the day has a typical, people use pipes as the basis of the buffer, and there's some magnesium in there, and some EGTA and so forth. As long as it's a standard, pretty ordinary microtubule buffer, then the tubulin subunits add on to the tip of the plus end the way they should. This buffer has, it's 20% DMSO, for example, and it's got double strength pipes than anyone would normally use. I believe, if I'm not mistaken, Heidemann just stumbled upon it by accident, but it causes these hooks to form. It's very abnormal, experimental scenario. Was there a question down here?
Is
this hook formation specific to the positive end of the microtubule?
No, it's along the side. It's all along the side, the lateral side.
So, sir, once you are isolating these microtubules from the dendrite or from the axon,
You're not isolating. You're taking a culture of cells and you are exposing it to detergent in the presence of this buffer and the extratubulin from the cow brain. And it's forming the hooks on the microtubules in the axon and the dendrite, flat on that culture dish. You can also do it, say, in a nerve that you dissect out of an animal. So you're not isolating.
there is no reorientation of the microtubules.
Well, no, there shouldn't be. No, I can't imagine how that would happen. So,
Yeah, I'm sorry to interrupt, but I think this is a relevant thing. And I was wondering, I looked at the literature recently in the bipolarity, and I think there's pretty good evidence that as you look along a dendrite, you have bipolar microtubules towards the proximal side. But as you go distally towards the tip of the axon, you have more unipolar microtubules, which are the same as the axons. And part of the reason why I think it's true is because we're just playing around looking at organelle transport and dendrites. And when we look at proximal dendrites, there's tremendous back and forth movement. But if you look at distal dendrites, you have fewer vesicles going, but the ones that are going always move persistently. So this is kind of indirect. I was wondering if you can comment. I mean, I think we need to incorporate that into the models we have here. And I was wondering how you think about it.
Well, one of the things that I wanted to mention either now or at the workshop is really to accentuate Peter Hollenbeck's point about how textbook these observations have become, when in fact there are very few descriptive papers actually on this. I published this initially in 1988 on cultured rat hippocampal neurons. I did the developmental study in 1989 with this distance effect that you're talking about. Then a couple years later, I reported similar findings on rat sympathetic neurons. There were a couple of in vivo papers, one by Paul Burton on frog mitral dendrites. He didn't see the length effect. There was another culture work paper that Kathy Collins did a few years ago, and some more recent stuff that I've been publishing. And then you have the Drosophila work that I mentioned. There is not an extensive body of descriptive data on this phenomenon, and there should be, because we are actually taking a rather small body of data and extrapolating it. For example, there was a study done a couple of years ago where people used this very interesting new technique, second harmonic something or other, and they're able to look at a brain and look at some sort of microscopic effect that would indicate bipolarity, mixed polarity versus uniform polarity. And they thought that, at least in certain kinds of dendrites, it was more uniform. Was that distal dendrites? I'm not sure. If it's more uniform, does that mean it was more plus end distal, or does it mean it's more minus end distal? You don't know. I mean, in the insect dendrites, it's much more minus end distal. I have a feeling that what's going on here is that there are different recipes, different proportions of minus and plus end distal microtubules, distributed differently in different kinds of dendrites, in different situations. That those recipes, those combinations, make these dendrites different from those dendrites. Short, stout tapering, a lot of branches, not branches, whatever the case may be, it's because of this recipe. I'm guessing.
You could even envision changes in that being part of the ontological history of a given neuron's dendrite.
And the plasticity moving
right,
forward.
Activity dependent.
Yeah. So I really like that idea, and I like the fact that your data are consistent with mine, because I was real young when I reported this.
Question.
Yes.
What do you mean by saying? What do you mean by saying these EBP1 proteins, they bind to this assembly microtubule placent, and so then what the other placents are doing? I mean, are they not undergoing polymerization or what?
The plus end, if it's undergoing one of these rapid bouts of assembly, like we talked about with the dynamic instability, then the plus tip will be associated with it. If that plus end starts disassembling, there's assembly, disassembly, assembly, disassembly, that comet will disappear. In other words, you will see that comet when the microtubule is assembling, but you won't see it when it's disassembling, because the comet is only present when it is assembling. There are cases in which a microtubule may be neither assembling nor disassembling, or maybe assembling rather slowly, not undergoing one of those big bouts of rapid assembly, and you would not have a comet in most situations. So you really only have these comets on a fraction of the microtubules. That raises a really interesting point. The hooking and the comet techniques, are they visualizing the same microtubules? Because they're both sampling techniques. When you're doing the hooking, you can really only interpret neurons, axons, dendrites. They have about 10% of the microtubules hooked. And someone could come along and say, well, how do you know that those 10% are representative of the rest? Maybe certain microtubules hook better than others. And then you might be saying, as you just did, there are certain microtubules that the comets reveal, the more labile ones. Well, someone could come along and say, you know what? I think that stable, really stable microtubules hook better, and really dynamic microtubules comet better. And it's true that these techniques may reveal different populations of microtubules. So this is yet another thing to keep in mind as you move forward. Keep in mind that your techniques are usually sampling techniques. And there have been historically in biology, I won't say errors made, but there have been, people have interpreted findings that are not broadly representative for that particular reason. In the recent paper we published with this, we always find, for example, in dendrites, when we do the hooking, the comet technique, we always see more plus-end distal microtubules with the comet technique than we see with the hooking technique. And I think what that must be telling us is that more of the plus-end distal microtubules in dendrites are dynamic and revealed by the comets. Was there? Yeah.
Can the axon discriminate between different isoforms of tubulins?
There are about 6 or 7 different primary gene products for alpha tubulin and a similar amount for beta tubulin. Beta 3 tubulin is the neuron-specific isotype. I think that whatever isotypes are available, that I don't know that they can be discriminated amongst. I think whatever isotypes are available will assemble into microtubules. But developmentally speaking, for example, older neurons will synthesize a different complement of these isotypes than younger neurons, and they can influence their dynamic properties.
Does the ratio of the microtubules with a particular polarity, like minus n towards the distal, does it changes in the dendrites, and if yes, how does it change?
Well, that really goes to what we were talking about just a moment ago, that developmentally, regionally, I think they do change. And I suspect that those changes are really awfully important. But the body of literature, the body of known data, is actually rather scant. There's not really sufficient body of data. Let's say I wrote a grant. Let's say I wrote an NIH grant saying, I want to spend the next 5 years of my life surveying dendrites and correlating microtubule polarity patterns with morphology and organelle distribution. That grant would be triaged. I would not be given $1 million to do that because it's not mechanistic, it's not really hypothesis driven, it's 100% descriptive. So that's one of the reasons why this really important work just doesn't get done, because people are always thinking about moving ahead mechanistically. And I'm the biggest advocate in the world for moving ahead mechanistically, but I also think there's a big body of information that's not known and that should be known, that a lot of assumptions are being made that really the scientific community could benefit from having that kind of knowledge. So this is a movie of these comets. And they're beautiful. Making these comet movies is a lot of fun. And you get a lot of information. You can see that this is a growth cone. And you can see that in the growth cone, most of them are coming out toward the periphery. Some of them you might kind of see them going kind of laterally. Some of them might be pointing a little bit more backwards because things get a little mixed up in growth cones. There are a lot of these microtubules, they're actually plus and distal, but they loop around. Some looping in the growth cone. But you get a lot of information from EB3 comets, depending on what experimental situation you're looking at. You can measure the duration, you can measure the speed, you can measure a lot of different features of these comets. So very data-rich studies that can be done with this. So,
I have one question.
yes.
So we see that the growth cone is moving, as in the active movement is there, and after that the microtubules are following somehow.
Yeah.
What moves those microtubules in that?
That's really been the question that has driven me, not since I was 4, but, a little older. My issue has always been what's organizing the microtubules, what's driving them forward. And this is also kind of an interesting story if you look historically. Maybe I'll talk about it a little more at the workshop. But it's really good, I think, to have passion. And students here, I just love the passion that you're displaying. It's wonderful. And I always had it within me as well, since I was a graduate student anyway, to understand how microtubules are organized, how they're driven into axons, dendrites, growth cones, and so forth. That was always my big issue. What mechanisms are organizing these microtubules? And there was a big controversy for a very long time about whether microtubules themselves actually move. And to cut to the chase, what I think the answer to your question is, although I'm not here to minimize the contribution of microtubule assembly dynamics, the thing that I think is most fundamentally important for setting up these microtubular arrays is the transport properties of the microtubules. In other words, these microtubules, once they come off the centrosome, they're moving themselves. They're moving themselves. So these microtubules are moving through the cytoplasm. And they're moving, say, with the plus on leading or the minus on leading. And the nature of their movement is all important. And I certainly didn't come up with this idea, but I thought it was enormously appealing. And I like it at so many levels. One of the reasons why I really like it is you think about fundamentals of cell biology. Usually cell types don't just concoct a novel mechanism. They don't just pull something out of thin air that never existed before, but they capitalize on themes. In mitosis, for example, microtubules move apart. And the idea that microtubules are moving into axons and dendrites, to me, is highly satisfactory. At an intellectual level, highly satisfactory. And some of the early pioneers in axonal transport, my postdoc mentor, Mark Black, was one of the luminaries in this early work. What this work would indicate in a very indirect way is that tubulin is transported down the axon at a slow rate compared to proteins that are associated with vesicles. And they call it slow axonal transport. Slow component A specifically is the movement of the tubulin. And what these early authors propose is that the microtubules are moving. That makes perfect sense. So the motor proteins would engage the microtubules and move the microtubules, and doesn't that make good sense? Well, the whole thing met with a lot of controversy that raged for decades, with a lot of people saying, nobody ever saw a microtubule move. You don't know if microtubules really move. It doesn't make any sense if they move. I think free tubulin is moving. And then someone would say, well, no, it's not free tubulin. It's some intermediate form that no one's ever seen, but I think it exists. And this would go on and on and on. I mean, this was before the days of PubMed and computers and all this, that every month I would get an issue of the Journal of Cell Biology. It was before Nature Cell Biology. And I'd open up Journal of Cell Biology and say, oh no, here we go again. Another paper saying, microtubules don't move. Microtubules don't move. I can't see them move in my lab, so they don't move. And then I would publish a paper saying, you know what, I think you're right. And say my reasons, and on 2 more papers, they don't move, they don't move. And these are very big labs. So this was a raging controversy. And this is another lesson, I think, for students. And really to again accentuate Dr. Hollenbeck's point, that just because a lot of powerful people believe an issue is resolved, doesn't make it resolved. And sometimes people have blind spots. And sometimes people have territory to protect, legacies to guard. The very idea that microtubules may move themselves, I think makes really perfect sense. Because if I had some little kid up here with me, I took that kid, I just threw it across it. Fine. You know what Arnold Schwarzenegger is? Big actor. If I had Arnold up here and I was trying to throw Arnold across the room, you know what's going to happen? I'm going to go across the room. Because all motion is relative. So, if little things can move on microtubules, why can't the microtubule move? Say, if the little thing, if the cargo wasn't a little vesicle, if the cargo were something big. So what if the cargo were like a longer microtubule? You got a short microtubule, you got a long microtubule. The short one moves. What if the cargo was the big old actin side matrix? The little microtubule would move. You're not going to move the actin side matrix. All motion being relative. And this really also is where this conservation of theme comes in. The mitotic spindle, mitosis, the most fundamental of all microtubule-related processes. And that involves a lot of sort of mechanisms. Nucleation from the centrosome, assembly, disassembly, but also there are a whole bunch of microtubule-related proteins that are pushing and pulling and forming the spindle and then dividing in half spindle as mitosis progresses. And I'll tell you another story. I was an assistant professor, and I shouldn't have, but I pretty much ignored the mitosis literature for a long time. I figured neurons don't undergo mitosis. Why do I need to bother with mitosis literature? And I always thought about, on my day of retirement, if by retirement I could discover the motor protein that transports microtubules, I would have a highly satisfactory career. And that was also before the days of sequencing the genomes. Pretty soon you run out of motor proteins, they're all discovered. It was also, at the time, people hadn't visualized microtubule transport yet, and we're thinking about slow motor, slow axonal transport. And there's no known motor proteins that are that slow. But I wanted to know, are there any motors, that could potentially transport microtubules, even just as a theme, even as just a proof of principle? So I had my graduate student, who's now actually a very accomplished scientist in the mitosis field. I had him do a lab presentation. Say, just find out about motor proteins and tell us about motor proteins. So he started to talk, and he put on the board, he put a microtubule, and he said, okay, so motor proteins, they have a cargo domain, they have a motor domain. And then he said, so there's certain motor proteins that are in mitosis, and they don't transport vesicles. Instead, they transport microtubules against each other. I thought, what? Can you say that again? It had been known for 20 years that there are motor proteins that move microtubules. I was that little, actually, 20 years before that. And so, every time we've looked for one of these mitotic motor proteins, these motor proteins that supposedly are only in mitosis, we always find them in neurons. And these motor proteins, kinesin 5, kinesin 14, kinesin 6, kinesin 12, they don't transport vesicles, they transport microtubules. This movie really changed everything. And this movie was reported by Tony Brown. And what Tony did is a very simple experiment. It's a photo bleach experiment. He expressed GFP tubulin. You see, in the past, and this is another good example for students, in the past people have done this photo bleach experiment in a very different way. They express GFP tubulin or microinject fluorescent tubulin, make a little tiny, tiny bleach zone, and then they wait 5 minutes, come back, and they say, oh, that bleach zone didn't move, because they figured that if all of the microtubules are moving slowly down the axon, they should see the bleach move. Well, what Tony does is a really long bleached region. This whole thing is the bleached region. This is the actual image. This is a subtraction image to clarify. And he saw microtubules moving through the bleach zone. Not the bleach zone itself moving, but microtubules moving through the bleach zone. And also, he took the images every few seconds, not every 5 minutes. You see, what the people did who performed the experiments prior to Tony Brown is they were going on a lot of assumptions. They were going on the assumption that all the microtubules would move together and that they all move slowly. It's like, let's say, if all of you folks were walking down the hall, holding hands slowly, and I looked out that window every 5 minutes, I'd see you. Now let's say you were all crowded at the other end of the hall, and one at a time, ran down the hallway. Then I looked every 5 minutes. I wouldn't see you. These were some really important big labs that reported over in Journal of Cell Biology that microtubules don't move. When Tony did these experiments, he was at a little, small, country school.
Ohio State.
No,
Ohio University.
First.
And he did these experiments, and they really turned the field on its ear. Because in one fell swoop, he not only saw microtubules, he actually was neurofilms initially, but he also solved the mystery of why those other guys never saw them. Because in actual fact, what's going on here is these microtubules are moving quickly, but asynchronously. And not very many of them move at one time. So all those biochemical studies, it's the same lesson for dynamic instability. All those previous studies were an average between a very small number of infrequent movements and a whole lot of non-movements. Average it all together in a biochemical experiment, and it looks slow. But if you look individually, you see a very small number of infrequent movements that are fast. And this really tells us a lot. Now, a couple of other surprises. This movement is anterograde. 2/3 of these movements are anterograde. But 1/3 are retrograde. So the microtubular transport, people had assumed was only forward. It's not just forward. Forward and backward. Secondly, only really short microtubules move. That was an interesting observation. And this really goes to the question I think Sanja asked about microtubule lengths. You know, most microtubules really have to be quite long in the axon because their jobs are A, to sustain morphology, to bear compressive forces, and then secondly, to be the substrate for these organelle transport events. Those microtubules really don't move. At least we haven't seen them. The short microtubules move. So, in order to get microtubules to move, they've got to be relatively short. That's where the severin proteins come in. So what I'm interested in anything that will help organize microtubules. And there are proteins that regulate nucleation, assembly, disassembly, bundling, all sorts of things. But I'm mainly interested in this microtubule transport. The reason why I'm interested in the microtubule transport is that I think that's how you set up these polarity patterns. In the axon, you want all the microtubules to flow into the axon with the plus end leading. In dendrites, it's two steps. First, all the microtubules flow into the plus end leading, and then later, as these processes become dendrites, a second wave of microtubules are introduced with the minus end leading. Question?
Is there any difference in the protein proteins which recruit the most to the plateau and the microtubules? Is something you know about it?
I think Scott probably knows more about that than I do, so maybe you can direct that question.
Well, I think it's all speculation at this point in terms of there are perfectly good hypotheses and models out there. The number of real
So why don't we just move on, because there's so many questions to entertain over lunch and dinner. And so the model that I like is called cut and run. Cut is the severing of microtubules into short enough pieces that can actually run. And the purpose of the run, the movement of the microtubule, is really twofold. One, set up the polarity orientation, and secondly, it's the means by which tubulin is introduced into axons and dendrites where it's not locally synthesized, at least not very much. So you've got to get the tubulin down there. So once you have these short microtubules flowing into axons and dendrites, probably what's happening is a lot of those short microtubules are becoming longer, they're adding subunits, and they become stationary. Other of those short microtubules are disassembling and yielding their subunits for their neighbors to get longer.
Whether these microtubules can join among themselves apart from like heterodimers, whether these microtubules as such they can join.
Or whether they can fuse, neurofilaments can. I don't know about microtubules. I don't know about microtubules. I sort of doubt it, but I'm not sure. But Tony has also shown that neurofilaments can fuse. They can.
There's in vitro evidence, old papers from Murphy.
Doug Murphy, yeah.
Microtubules. I don't know of any within a live cell, in vivo experiments that indicate any other.
In vitro, they have seen
in vitro, Doug
and
Murphy.
then dealing with microtubules has been demonstrated in vitro in a tube.
So I think what probably is wise is if I try to wrap this up a little quickly because we have other speakers. So I'm going to talk, I think it's 8 p.m., first or second day of the workshop. And we're going to talk more about this, second day. So we'll talk more about it. I'll try to go a little quickly and just hit up on a couple of points. One of the things I've really been interested a lot lately is what is the point of the retrogradely moving microtubules? Why do they even exist? I just told you two good reasons for the anterogradely moving microtubules. Well, what about the retrogradely moving microtubules? Why are they there? And my hypothesis, which is very difficult to test, my hypothesis is, what if, just imagine, what if those minus end distal microtubules represent flipped, maloriented microtubules that somehow got corrupted, somehow they got flipped. Maybe during a severing event, they got too short and they flipped. They're no good. You don't want them. Because if they elongate the axon, you're going to corrupt that microtubular ring. So those occasional minus end distal microtubules that popped up as revealed by the comments, you don't want that to get out of hand. If that gets out of hand, you're going to ruin the axon. You're going to corrupt the microtubular ring. So what goes on is that the short microtubules that are the wrong polarity orientation, they're on their way out. They're being cleared. So the retrograde movement has a very, very important role to play in preserving the uniform polarity orientation of the axon. That's a hypothesis.
So which regulates the splitting of microtubules? Which factor regulates this?
It's not that you want it regulated. It's something that I would put it like this: that it's slippage in the system. That if the microtubules during a severing event get real short, they might be able to accidentally flip. Or say something like you form a branch. When you form a new branch, you have a lot of severing, and they're kind of mixed up. Then some of them just accidentally flip. So it's a normal kind of thing that happens that creates a little noise in the system, a little error. Is a good word. It's a little error in the system. So you've got to have a constant mechanism to clean up the trash, to correct the error, to clear up the bad guys.
So, sir, according to this hypothesis, there should be polarity-specific motor.
Well, if dyne is the motor that's transporting the microtubules of plus and distal normally down the axon, once they flip the other direction, all you need is dyne. All you need is the same motor, because now it would still move with plus and leading, which is move the other way.
So, sir, according to this, there is equal probabilities. Something will come from this side, something will go from this side. So this hypothesis, it will go both the ways.
Well, if the microtubule only moves with the plus end leading, right? If it only moves the plus end leading, then if the microtubule is pointed toward the tip of the axon, it will continue down the axon properly. If the microtubule got flipped abnormally, now its plus end is pointing toward the cell body, and all you need really is the same motor for that flipped microtubule to go toward the cell body. I'm not saying it's a single
What about
motor.
the error in the other portion of the neuron? What about the error in other portion of the neuron? If there is the same flipping, that will come to that this way.
You're saying in other areas besides the axon? I don't know. I don't know, because in dendrites you naturally have a non-uniform pattern. So dendrites would be another challenge altogether.
But when the motor is carrying microtubule, disoriented microtubule, it's carrying as a cargo. So why does it matter what is the?
I don't think it is carrying it as a cargo. I think it's a sliding filament mechanism where the short microtubule is not the cargo. The short microtubule is interacting with the motor domain. The cargo domain is interacting with the stationary microtubule, the long microtubule. If you're correct, you're absolutely correct. If it's a cargo mechanism, what does it care? It wouldn't care. So it's a sliding filament mechanism. It's the directionality of the motor. Maybe I can draw it for you later.
But in that case, it should be the microtubule should be big enough to move as a track.
Well, big enough, I suppose, but what does big enough mean? I mean, the microtubules that are mobile are generally about 7 to 10 microns long. So I hope that's big enough. So many questions, yeah.
So, if the microtubules are moving in one direction, that would imply that the array of the motors are, in same conformation. They have some kind of polarity, the array of these motors.
Yeah.
So,
yeah.
It seems unlikely that they will arrange by themselves. There must be some mechanisms to arrange them in such a way so that they are in same.
Okay. Real quick, anyone else have a real quick point? Over here.
It's, as you said, 33% of the times the mitochondrial moves retrogradely, but you see the negative minus and distal microtubule arrangement only once in a while. So I would not expect that to be a major reason for having the retrogradely moving microtubule.
Well, the way that I look at that, I don't know if it makes mathematical sense, but the way that I look at that is that it's because of the retrograde movement that the neuron is able to keep down the number of longer microtubules that have the wrong orientation. So it's a very active mechanism. Let's say you have a long microtubule that's the wrong direction. You chop it into several pieces, and all those short pieces move retrogradely. Remember, the short microtubules will not have a comet. It's only the long microtubules that have a comet. So keep in mind the comet, the percentage may be a little deceptive in that sense.
Does that mean that the proportion of retrograde moving microtubules will be different in terms of how far is it from the soma?
That's an interesting point because I've been pondering how to test this hypothesis. It's just a hypothesis. It's just a hypothesis for now. And to be able to understand, to know the polarity orientation of the short microtubules, tricky. It's very difficult because you have to see it moving at the same time as know its polarity. How are you going to do that? I've got a few ideas, but they're very tricky. So I thought about, well, if the hypothesis is correct, if we look, say, with a hooking technique, very, very proximal, would you see more of them maloriented, very, very proximally? So kind of thinking similarly to you on that, but we'll see. So this all has to do with the polarity of these short microtubules, like I said. Our older model was that all the short microtubules are plus and distal. We didn't even think of this. The newer idea is that the microtubules moving anterograde are plus and distal, but the microtubules moving retrogradely have the opposite orientation. Again, untested. We need to be able to test this somehow. We think this might happen, as I mentioned before, say by severing of them. When you get axonal branch formation, the microtubules get severed in short pieces, and they get mixed up. So any sort of plastic event that happens might cause this mixing. And here's the idea I think some of you guys may find attractive. What about a disease scenario? What about a disease scenario where you have axon degeneration? And I think any sort of toxic misfolded protein, any sort of abnormal destabilization of microtubules or abnormal breakdown of microtubules could very well result in a corruption of this microtubule polarity pattern. Let's just say the retrograde transport doesn't work so well. Let's say that doesn't work. So you don't have a clearing mechanism. So if you fail to have a clearing mechanism, or if the clearing mechanism isn't powerful enough to clear the corrupted microtubules, you're going to start having problems, say in Alzheimer's disease, or Parkinson's, or Huntington's, or whatever the degenerative disease is, where you might have pockets, regions of microtubules that really are getting the wrong orientation and allowed to elongate. And then you're going to have issues, like you're going to have abnormal traffic jams. You're going to have accumulations of organelles. Things are going to start to go awry because the polarity patterns have been corrupted. Now this is all hypothetical for now. This has to be looked at too. Remember I said that not enough has been done to look at normal microtubule polarity patterns. Well, nothing has been done to look at microtubule polarity patterns during degenerative diseases, and I think it has to be done. And I think this could really be rather important for diseases. I think also, if you want to get your grants funded, you better think about disease.
Yeah, a question. So what do you think happens in case of regeneration? Do you think that after regeneration from distal region, if you take the cross section, you should see mixed polarity of the microtubules?
Regeneration?
Regeneration. Suppose you have cut, because you're telling that
cut axon?
Cut axon. And then what if the microtubules which are coming back, if they have different polarity as compared to the anterograde, which are moving? So
I suspect if you have a cut axon that is to regenerate, then it's initially the microtubules get kind of mixed up, but they've got to reestablish themselves. They've got the mechanisms have to kick in to reestablish themselves. You know, another thing that's related to all of that is people want to use, say, Taxol to treat regenerating axons, to enhance generation. Taxol is a microtubule stimulatory, stabilizing drug, and that could create abnormal polarity orientation. People want to use drugs like Taxol to treat Alzheimer's disease. And fine, I think that's going to stabilize microtubules and preserve them. But I think that the Taxol is also going to create non-uniform orientation. What else is the Taxol going to do in terms of, say, the EB3 commons? If you stabilize your microtubules with the Taxol, what's going to happen to EB3 commons? They won't be there. You won't have them. And those EB3 commons are not just God's gift to scientists. They have a function. So this is why when you consider disease and therapy, all this basic science comes to play. Are you standing here because you want me to shut up?
No. I just want to, this discussion is really nice and lively. But just because, we have to finish today, there are many talks. So please, hold your questions till Peter finishes his points.
I really
discussions later on. There will be a discussion session which Shubhajit and Scott is going to order.
Also, I must say, in the workshop, in
this discussion, but
in the workshop, I don't have anything planned, so I will just continue this in the workshop. I don't have any. I was just going to talk, so this will work out very well. So I can just part 2 coming up. All right.
So next speaker is John Morrison.
Thank you.
And John is going to talk about myosin, which we haven't yet really
yes, please.
talked about, and we'll see how myosin is also involved in
that would be nice.